Pynapple. Just out of curiosity, before coming here, how many people have heard about Pynapple already? Oh, wow, okay. We are geared for success here. Okay, so first question is where does Pynapple comes from? The package is recent, but the history behind is quite old, actually, and I will describe that. I will describe it by showing these two experiments. So those are two experiments or two data sets I've worked with in my life. One on the left is some sort of electrophysiological experiments where you basically put electrode in a rat and you ask the rat to sleep. And then you record from the cortex, from C1, and you get some nice units here. And you get a nice spike count. And then the point of this study was to show that whenever there was a dip in the spike count here, there will be a sharp wave ripple in the hippocampal LFP. Okay, so that's one type of experiment you can find in neuroscience. And on the other side of this double arrow here, I show another type of experiments, which is, I would say, more structured. In this case, you have a set of stimulus, a set of action, and a set of response. Okay, so then you can do a lot of reinforcement learning type analysis. But you will agree that this type of experiments here is, I would say, more structured. Here you can really compress the data set into a few sets of arrays, an array representing the stimulus, an array representing the action, and an array representing the reward. Okay. On the other hand of this arrow here, this is much less structured and it's more difficult to deal with. You have a bunch of time series, and if you have done sleep research, you know that the rats never sleep the same amount, sometimes half an hour, sometimes 3 hours. He wakes up. So those are a lot of uneven duration of time series. You have a different number of units. You have different intervals between the events here. So to deal with this kind of experiments, which are much less structured, the first author of this study, Francesco Battaglia, made a few decades ago a toolbox in MATLAB called the TS toolbox. So I think that was in Bruce McNaughton lab like a few decades ago. And few places in Europe and North America later, and a few decades later, I was a postdoc in Adrian Perach's lab, and when I arrived in his lab, he gave me the TS toolbox. Adrian Perach was the PhD student of Francesco Battaglia, so he gave me the TS toolbox. There was no documentation, and I prefer Python in general. So thankfully, Francesco Battaglia also made another version in Python called NeuroSeries. And from NeuroSeries, I started writing code around it. And then soon enough, I was writing code for my whole lab. And then after a few years, we decided to make Pynapple out of it. And now I'm writing code for everyone. Okay, so that's more or less the story behind Pynapple. That's just to give you an idea that, in fact, the package is recent, but the ideas behind it are very old. Okay, they are as old almost as the first people that were doing electrophysiology. Well, not that old, actually, because it will be very old. Okay, so now another question, when do I need Pynapple? Okay, that's also some confusion sometimes about people that are being introduced to Pynapple. When should I use it? So I will show you another arrow here, and this time it is a timeline of an experiment, okay? You might find yourself along this kind of arrow here for like, from the moment you collect the data, if you are more like on the experimental side, to some pre-processing pipeline that you might apply to your data. So here we just learned about spike interface. Down there to some more advanced analysis that you basically are maybe on the computational side and you are collaborating with someone, you receive the data that have already been processed and you want to apply some manifold analysis or some modeling analysis, okay? So very likely you are on this spectrum, and Pynapple basically lives here. It lives between the pre-processing and the post-processing aspect of data analysis of neuroscience. Once you have done your spike sorting or your cell sorting with Caimon, you basically can take your data, and this is when you usually do this sort of analysis where you start to put your data together, you check if the time alignment is correct, where are the good units, I'm starting to segment the recordings into like epochs of like, okay, this is the epochs of noise, this is the good epochs, and so on. So this kind of data wrangling that you usually do after the pre-processing, where you start to extract the good aspect of your data, basically. You also do some tuning curve computing, you do some cross-choreogram, you check for the quality of your data. So all of this data wrangling can be done with Pynapple. It can help you align your data. And then after that, usually what you do is that you call like another package, for example, scikit-learn or something I will talk about also after, for doing some more advanced analysis where you want to fit a model or you want to fit like complex representation of your data. Does that make sense about where Pynapple lives in your pipeline of analysis? All right. Okay. So that's what I will describe now. And here I show in these boxes how from like the main idea behind Pynapple is that from all sorts of experiments, either if you have like patch clamp, electrophysiology, calcium imaging, all the way to like monkey recordings, all of these experiments generate time series, timestamps, and intervals, right? Different name for them. But the idea of Pynapple is that instead of having objects that will basically match the exact name of your data sets, you have a few number of objects that are named very arbitrarily. I can represent anything, basically. That's the idea. So all this experiment will be represented by a few number of objects. I will describe that now. That makes sense? Okay, we're going to do a deep dive into Pynapple, and then I hope it will make more sense about what it can do. All right. So let's start. First things we usually generate in neuroscience are time series data. And in Pynapple, they are represented by three types of objects: the TSD, TSD frame, and TSD tensor. And the first element, the most simple one, is a one-dimensional time series. So here I show, for example, patch clamp recording. So you record the membrane protection of a cell. Okay, this is a one-dimensional time series. And so in Pynapple, you can represent it as a TSD object. So if we import Pynapple as NAP, which is a standard namespace we have, NAP.tsd, you pass the time axis, which is at which point is your sample in time, your data, and you get this object TSD, which represents like this. So it's very similar to Panda data frame, okay? It has the time as the first axis and the data as the second axis, all right? So very simple so far. That's one dimension. Under the hood, it basically leverages NumPy. If you are familiar with Python, you are very likely familiar with NumPy. So it's based on NumPy, and it's this very similar interaction. You can access the time axis. You can also access the value, and it returns the NumPy array. Okay, so nothing fancy here. We stay as close as possible to the what is standard in the Python analysis ecosystem, which is NumPy. Okay. But you gain many things in addition to that, and I will describe them along these talks: automatic handling of time units, epoch restriction, binning, time support, and so on. So there are some aspects of it that are more than just having a NumPy array that I will explain to you, because you might say, okay, why don't I just use NumPy, basically? Now, this was for one dimension. Is it clear so far? Yeah. So this was for one dimension. But sometimes you acquire data sets that are more than one dimension. There are two dimensions. So the example I show here is basically you record the position of your mouse in a 2D environment. So you have an X and Y position, okay, in your environment. This will appear like this in Pynapple. You will instantiate what is called a TSD frame object. So this is very reminiscent of Panda data frame. You know, Panda data frames are like column-based object, okay, that you can label either the rows or the columns. So it's the same idea here. It's called the TSD frame, where you pass the time axis, the data, and you can label the columns. So then you have a nice representation of your data, which are basically time here, my X column, my Y column, okay? So very simple so far. Now we went from one-dimensional to two-dimensional. Here the important aspect is that every time point here, every data point here is sampled in the same time point, basically. All right. Now that's for 2D. What about ND? What about the movies, for example? Well, we have the TSD tensor object. So I mean, the only example I usually think of is movies, because anything more than 3D, I don't know what people are doing with more than 3D, but maybe it exists, it's possible. But here I show what happens if you record a movie. You can also pass the time axis, the movie, and you get a representation of your tensor, for example, a 15 by 15 pixel movies, okay? And then, of course, with this, you can apply the same type of analysis that you can apply to all of these other objects. Now, that's 3 objects. I want to describe a little bit NumPy compatibility. As I said before, you can basically extract the underlying arrays as NumPy arrays, but you can also apply any NumPy function. NumPy very nicely exposes something to basically apply any NumPy functions to this kind of object. I will make it clear now. So, for example, if you have a TSD here, you can apply any numeric arithmetical operation, like adding anything to the array. You can apply another. You can append another array. You cannot append 2 arrays 2 time series together, because here you need to do a lot of verification for the time axis. Okay, if you append 2 time series together, how do you make sure the times matches perfectly? That is a very hard problem. So the solution was just to make it incompatible in Pynapple. Okay. But what I want to describe also is that if, for example, you have a movie here, and let's say I want to do the average of my movie along time. So I want the average frame, okay? In Pynapple, it's possible. You can just say np.min my tensor axis equals 0. If you are familiar with NumPy, this looks very intuitive, okay? Along the first axis, which is time, give me the average pixel, and then you get a NumPy array. So here you go from Pynapple to a NumPy array. But you can also do something cool, which is now I want the average of my pixel along the second dimension, so this dimension here. And what do you get? You get a TSD frame, because Pynapple catches the call of the NumPy function and say, okay, am I spitting out something that preserves the time axis or not? If I don't preserve the time axis, I spit out a NumPy array. If I preserve the time axis, I spit out a Pynapple object. And here, in this case, you keep the time axis, but you get a new object, which is a Pynapple TSD frame. So you lose 1 dimension, and then you get your average of each pixels. Okay. So Pynapple knows how to deal with NumPy function scores. Okay. So that makes it, when you write your code, it's very easy to then write code that looks very neat and clean. It's clear? All right. No question. Good. You can index a Pynapple object like NumPy array. You can index, like you can slice, and then you can do even more complex slicing. Okay, so just to think of it as a Pynapple object as a NumPy array, and it should work out of the bat. You can do array concatenation provided that the time axis basically don't overlap. So this time point here is 9, this time point is 10, so this should work. If you do the opposite, this should not work, because in Pynapple, the time axis is always ascending, so that if you have a time point that reverts, it will show you an error, okay? So there are those are the type of checks that are inside Pynapple that helps you basically catch errors sometimes. So maybe you have like some axis, some time points are not sorted. Pynapple will show you an error saying, hey, the time points are not sorted, okay? So that's the reason why you can use Pynapple, is to catch this kind of errors. Okay, makes sense so far? So we have 3 objects, all right? Okay. And in neuroscience, we often capture data sets that don't have data with them, but just have times. So time series of data, the timestamp object. And the example I show here is a mouse on a wheel, and then you get a reward, basically like a drop of water. And so you record at which point is your water delivered. So this is just a time, okay? There is no data associated, okay? So for that, in Pynapple, you have the timestamp object, which you just pass an array of time, and you get this simple representation of like time without any data. But sometimes we get time with the data from multiple channels at the same time. And here, of course, I'm talking about population of neurons. So if you recall, for example, brain activity, you get those units in time. So every row is a unit here. And you record these spikes. You can group them in Pynapple in what we call a TS group. So here, those times were recorded together. So you can do group of timestamps. And this is a TS group object, all right? So here, data is a dictionary. I will show after how to create this. But basically, when you instantiate a TS group object, you get this nice table where every row is a unit here, or it can be whatever you want, actually. And it's a TS group, so you can show whatever you want here. Of course, the most useful case is population of neurons, but it can be anything else. And it gives you the rate of this particular object units here, okay? I will dive a little bit into the TS group object. I call it population analysis made easier, the TS group object. Okay. So under the hood, the TS group object is a dictionary. So if you are familiar with Python, new dictionaries are basically a mapping of key and values. Okay. So Pynapple leverage that to basically create the TS group object. So if you pass a dictionary of like key, which is the index of the neurons, to a set of timestamps. So here, for example, I show an example of 3 neurons recorded together. It will show you a nice table, okay? Now, the cool thing about it is that you can index your table very easily. You can also apply one of those Pynapple functions that applies to those time series object or timestamp. So one of the things that we usually do when you have like a population of like neurons is to count the spikes. You want to bin the spike train, okay? Because that's the basis of doing any advanced analysis is to bin the spike train. So you can do it very easy in Pynapple. And now I would like to highlight something about one reason why you would like to use Pynapple. That's think about sharing your code. We talk a lot about sharing data. Okay, sharing data is important. What about code sharing? And if it happened to you already that you have joined a lab and then you receive the code from the postdoc that left 5 years ago, then it might be like a bunch of spaghetti code with people doing this kind of very simple calls of like simple analysis, just binning a spike train. That sounds trivial, but maybe he did with a call of histogram, maybe he did a double loop, maybe you don't know what he did basically. But then if you use Pynapple, you have like a very simple way of structuring your code, where if a new student reads this and says tsgroup.count, I say, oh yes, okay, here he bins the spike train with a 2 second bin size, okay? So then when you share your code, and if you both use Pynapple, it's much easier to read someone else's code, because then it's a call of the same function for very simple things, okay? That's one of the reasons Pynapple can be useful when it comes to code sharing. All right? Now, that's it for the TS group object. One last object we need, and then we're going to be complete, which is the interval set. Interval set, of course, when is the start and the end of my experiment, when is the start and the end of my stimulus, when is the start and the end of the REM sleep, whatever, okay? So we need to represent that. So we have the object called the interval set object. And here I show an example where we have some gratings, okay? And it appears as a table here where you pass a list of start, a list of end, and it shows you like the start and end of the epochs, all right? Now, I call it manipulating time. So here are a few examples I say. For example, sleep, wake, where the start of sleep, the end of sleep, the start of wake, the end of wake, okay? When is the start and end of the stimulus? When is the start and end of the leak, okay? So you can represent anything here. The most intuitive way of representing intervals is a table like this. So this table here will basically translate to this object here in Pynapple, where you have the list of start and the list of ends, okay? So very easy to create and very easy to read. I would like to add that the epochs are strictly non-overlapping. Two epochs cannot overlap, and epochs are always sorted ascendingly, okay? They cannot be reverted. Now, here is one of the core functions of Pynapple, which is something that you do, like if you develop this kind of analysis pipeline, you do 1 million times. I want the data that are in these particular intervals. I want only the data during sleep. I want only the data when the animal is turning right. I want only the data when there is this particular type of stimulus being presented to the animal, and so on. So this type of things are called restriction in Pynapple. So you want to restrict your time series, your timestamp, your spiking activities to those particular epoch. So there is a function for this in Pynapple, which is very central to the Pynapple ecosystem, which is a restrict function. So this interval set can be used to restrict your time series. So here, for example, you have a set of timestamp objects. Okay, maybe those are spike times, and those are a bunch of intervals, for example, 0 to 1 second and 3 to 5 seconds. And I want only the spikes that happens within those intervals, okay? So you can just do ts.restrict_ep, give me only the spikes happening within those two epochs. And as you can see, you have these spikes here and these spikes here. Okay, you can check quickly that is correct, right? It's correct. So this is a central function of Pynapple, the restrict function. And so basically, if you think your pipeline or data analysis pipeline as you take your raw data, I mean your pre-processed data, and then you kind of go down of like, okay, I'm going to select this and this and that and that. So you kind of like funneling your data into some, like the good aspect of your data. This is how you do it, by restricting until you get only what you are interested in, basically, okay? All right. You can also do some more fun stuff with interval sets. You can combine interval sets in different ways. So I'm just going to go quickly over this. So if, for example, you have two sets of epochs, you can do the union of epochs. Very handy when you say, for example, OK, give me every time the mouse going right while the queue is on, basically. Give me every time, OK, I have to find an example here. Every time there is this activity above this particular threshold and there is this stimulus on, basically. So this type of unions that you can do. Difference, for example, the classical one, remove every time there is noise in the system. You identify noise in your recordings. You want to remove them from your recordings. You can do the difference. You can do the intersection. So, for example, whenever okay, I'm always going to say the mouse going right on this happening, but you get the idea, basically. Sorry. So, okay, so that's some, again, think about code sharing. You go through your code, you read, oh, an epoch union another epoch. That's very easy to understand. If someone were to make their own custom function, they would probably not name it like that. Then if you were to look at the code 5 years later, it would be very confusing. So again, think about code sharing here. So summary, 6 objects to represent data. Now we are complete. With this, we can capture more or less all of the dataset in system neuroscience, I would say. Everything that generates a time series, basically, and that you have to basically do some data wrangling with it, all right? So the data wrangling does not stop here. We can do more than that. Is it clear so far? Yes? Am I out of time? No, it's okay. All right. So one thing we also generate a lot in neuroscience is metadata. So you're probably very familiar with this in NWB. A lot of metadata associated with the where is your probe, what type of intervals it is, and so on. So it would be nice to capture those metadata very easily. So in Pynapple, it's possible. So if I come back to my TS group example, here I say, for example, that these neurons were recorded from the thalamus, this one from C1, this one from cerebellum. I want to carry that in my object. I want to be able to show that in my object. So it is possible by basically declaring a metadata field where I say the location of those neurons are thalamus, C1, cerebellum, and it will appear like this in my TS group object. Okay, so every row is not associated with a particular metadata. Totally arbitrary. You can write whatever you want here. It will appear here. Okay, so we do not predefine the metadata. But now the cool thing is that if you receive the metadata, again, code sharing, if you define nicely your metadata and basically you share your code, if your metadata are well defined, for someone also reading your code, it's easier to understand what this person is doing. Oh yeah, he defines these variables and he filters and so on, okay? So you can do it for TS group. Yeah, so here, for example, let's say during my pipeline, I define a direction variable, then I can store it here. Also, when it comes to writing code, storing in one particular place your metadata, it declutter your code. Because like you generate those variables, you have to carry them through your code. And sometimes it can be very confusing. So then when you generate them, you associate them with the particular objects they are created with. Then you declutter your code. That's also an idea behind metadata. You can select, you can also create them on the fly, like many ways. If you think about a way of adding metadata, it's probably already there in Pynapple. So here is for TS group. So you can also do it for interval sets. So for example, here I want to associate the type of gratings I have for each epoch here. So here I'm going to say trial type left, right, left, okay? And I'm going to get this particular table where now there is an extra column here describing my metadata, okay? Also, TSD frame, for example, here, let's say I want to give a color to every column here, basically. So I can do the same, metadata equal blah, 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 blah. Okay, and now it appears like this. There is this extra column, extra row here, this associating metadata to this particular column. So you can add as much as you want. Yes? It threw you an error. It threw you an error. Yeah. There are some checks for that, yeah, internally. Okay, so yeah, you can add a lot of things, yeah, of course, from there you can do some selection, so you can access the metadata. I can always recover my metadata after I append them to the object. But the cool things you can do, of course, is to slice. So here I'm showing you an example where you have like 3 neurons. And let's say I want all the neurons in the thalamus. So here how you do it, tsgroup.location equal thalamus, and you get only the neuron in thalamus. Think again about code sharing. This is probably more easy to read if you have to read someone else's code than if you have to do it yourself, where you will create your own index and go through your own objects to slice your own population of neurons, okay? You can also do it like this, basically all the neurons. That's something I usually do in the beginning of all my pipelines. I don't care about the neurons that fire less than 1 Hertz. I want the good units. So I say, okay, everything below 1 Hertz, I discard, basically. I want only the neurons above 1 Hertz. And maybe I want all the neurons having this particular direction. Like I don't know what I was doing here. Okay? So yeah, metadata, think about it whenever you have to store something associated with your data, basically. Now, let's go for more data wrangling. The core function of Pynapple, there is a lot of them. I will not describe all of them right now. Just to give you some sort of summary table, you can go to the documentation page. The restrict I described, but you can do also the count, I described it. You can do value from, which is kind of way of basically mixing data between different time axes. You can do threshold, and there is more than that, all right? So from now, I think I will go to the Pynapple I/O. Is there any questions so far? No? Is it clear? Okay. So Pynapple I/O, how to get from our data to Pynapple? So that's a tricky question. And here is a slide from Spike Interface, which is the same as Chris. This is not what we want to do. This is the reason why we have NWB, I guess. But we don't want to support all of that. So very early on, I made the choice of not supporting all of that. I made the choice of supporting NWB. So you all know about NWB at this point. So I will go kind of faster above this. Pynapple supports NWB. So if you have an NWB object, I will now show you how to open it with Pynapple. Okay? Oh yeah, I should probably also skip that. I guess you've seen already that. Okay. Now let's say you have an NWB file, you already made your NWB file, or it's online, you can download it. And then if you give the path to your NWB, there is a function in Pynapple called nap.loadFile. It gives you an NWB object in Pynapple. And then from there, it's very easy, you get this table. So the idea is this one. There's a lot of information in NWB, but Pynapple is all about time series, timestamp, or interval set. Okay, so what it does is that it takes the NWB, it flattens it, and for each object in NWB, it asks the question, is it compatible with Pynapple? If yes, it proposes it to you as a Pynapple object. If not, it doesn't show it to you, okay? So then it basically simplifies the NWB by just giving you what you can load in Pynapple, all right? So in this example, in this particular NWB, there are this particular number of objects that I can load as a Pynapple object. And then if I do NWB, for example, position time support, I get my object from the NWB file, okay? So here in 3 lines, I've been able to extract the data from the NWB, all right? So that's just, like NWB has a lot of things. You can also extract everything manually, but here just a fast way to get from NWB to Pynapple object, all right? So you can try it on any of, you can just do pip install Pynapple right now, do this call, and you should be able to get started with Pynapple, all right? Any question with that? Yes? Yeah,
sort of
yeah.
On, off.
Yeah.
So there, for instance, how does it suggest that something is an interval set and something is a TSD? I'm imagining that most interval sets would be stored as like values where.
Yeah, but.
For instance, in my lab we have a thing that's like the lever on, lever off, and there are like 2 events, and one is a timestamp of the lever on, and another is like timestamp of the lever off. So
yeah. That depends on how you save it in the NWB file. Because NWB has some field name that are very specific, and I can't cite them by memory right now, but then some of them will automatically translate to an interval set. Some of them will automatically translate to a TSD frame. Like there is a mapping between basically NWB field name and the Pynapple object. Yeah.
So the NWB format has like conceptions like for this sort of thing, like when it's epoch.
Yeah, there are tables for that. Yeah. And and then Pynapple goes through them, and it recognizes there is a start, there is an end. So then I'm going to create an interval set. Yeah. And then he tries to be smart with the tags and try to give you, but sometimes you have to split them, but yeah, he tries to give you something. Yeah.
But you can also manually construct some of it.
Yeah. So then if you do nwb.nwb, you have access to the whole file, and then you can do it yourself, yeah. But here it's just like, okay.
Yeah, if I have like 2 TSDs like the ones I mentioned, there, I can just convert them into an interval set in Pynapple, for example.
Yeah. I mean, it always depends on how they were stored in the NWB file in the first place. So here the type will be inferred by Pynapple, but maybe something then you have to load it and do the conversion yourself. Yeah, makes sense. Okay. All right. I keep going. So, okay, so summary, middle slides. We have a bunch of data from all sorts of experiments. We can leverage NWB to get to Pynapple, and this is what we say during our workshops, like start by making an NWB file, because then it's very easy to go from the NWB to Pynapple, okay? There's one thing missing here. So we have our object, we have all our methods to do some data wrangling. We want to do some little bit of, how to say, not so complex, but still not so simple analysis. So there's an analysis module in Pynapple that I will describe now, which can do kind of, I would say, standard analysis in neuroscience. Yeah. I will give you an example here by saying, okay, let's say you have an experiment where you have a mouse running on a circular wheel here, and basically you record some data, and maybe those data appears like this for your experiment, where you have basically some LFP, okay, some neurons. You have 3 neurons here. You have some feature, which is the position of the mouse on the environment, and then you have some reward, okay? So this, maybe imagine you make an NWB file, you load the NWB file with Pynapple, this will appear as this type of Pynapple object in your environment, okay? There will be like a set of epochs here in green, the LFP will be a TSD object, the spikes will be a TS group object, the feature will be a TSD object, and the reward will be a TS object, okay? So this bunch of objects where you can get started with your data analysis, all right? Now, first question I can ask is, let's do some wavelet transform, for example. So let's take the LFP here. Sorry, what am I doing? Why is it the same thing here? Oh, yes, sorry. Let's do some wavelet transform, so which is possible in Pynapple. You pass the LFP, you pass a bunch of frequency, and I can examine what are the frequencies in my signal here. So you see a nice frequency here, okay? You can do bandpass filtering, okay? So very easy, apply bandpass filter, you pass the TSD object, a bunch of frequency, and then you get a filter version. So very useful if you want to detect some some high frequency event, for example, sharp wave, or if you want to look at the theta rhythm or anything else. You can do tuning curve. What are the firing rate of my neuron as a function of my feature? Okay, so we are in the process of renaming this function. So I will have to remember to send you an email when this is renamed. But yeah, you can compute tuning curves. Basically, you pass the spikes, you pass the features. What is the firing rate of my neuron as a function of the feature? So you see those 3 neurons here. You can compute cross-choreogram. What is the firing rate of my neuron as a function of another neuron? So here you have the neurons orange here firing as a function of neurons blue here. So 0 is the time of the blue neurons. And as you can see, the neurons orange increases firing after. You can do peri-event. What is the spikes in relationships to my particular reward time here? So I pass the TS group, the timestamp object, and I get a new TS group object describing the spike times here. Okay, you can do decoding. So for the moment, we only have Bayesian decoding. We are in the process of implementing template matching. But basically, if you pass the tuning curve, the spikes, the epochs, and the feature, you can decode the particular position, particular feature as a function of the spiking activity. Okay, so all sorts of analysis summarized here that are, I think, are already kind of extensive. You can already do a lot of this. You can almost do a publication with only this, I would say, if your data are quite rich. And just emphasis here. Okay. Is it clear? Yes. I went a bit fast here, but you can test them yourself. Like you go on the documentation, and there is a description for every type of analysis. All right. Make sense? Okay. So to summarize, this is the extent of Pynapple from NWB files to a few number of objects to a number of methods that allows you data wrangling to very simple data analysis in system neuroscience. Okay, and that's kind of the in and out of Pynapple. There is nothing more than that. It might extend in the number of analysis, but that's more or less it. Okay. Any question? No. Yes.
The last time I checked, there wasn't a lot of support for plotting within Pynapple. It was just kind of you output the data structures and then it's up to the user to plot it. Is that still the case?
Okay, I will come back to this later. I was torn between showing this or not. So sad because yeah, okay, I will come back to this since you asked. Okay, all right. I will show unfinished project, which I don't like to show. So I like to show unfinished stuff, but okay, I'll show it. All right. So just quickly, middle summary. I'm not done. Do I still have time? Yes, I have. Okay, I'll try to speed up. So pip install Pynapple in your favorite environment right now. It should work right away. We are on Twitter, on Blue Sky. There is a Slack if you want to ask questions. I would like to thank all the contributors now. I'm not done here yet. I want to talk about one more thing, which is this part here. Especially since we are NeuroDataReHack and you are probably doing some data analysis, you might want to do some modeling, okay? So it's not only me in Flatiron. I would like to talk about a project by my colleague Eduardo Balzani, who has been developing NEMOs for neural modelings, okay? So if you are more on this part of the spectrum of where you stand in neuroscience, you might want to hear about NEMOs, okay? So NEMOs is a modeling framework for system neuroscience, and the idea here basically is that, okay, for the community, when it comes to like modeling, you probably have used scikit-learn, but there is no like real standard implementation of classical models, okay? And that's the idea behind NEMOs, to provide standard implementation of established system neuroscience models. We can have a long debate about what is established system neuroscience models, but I would say the first one that comes to mind to people is really the generalized linear model. Who has heard about the generalized linear model? No, yeah, okay, few people, yeah. It's kind of standard. I mean, it's been around for a few decades now, and it's the one that works very well. So the one we have so far, we are in the process of doing more. So I will describe NEMO's more or less implementation. Since Eduardo Balzani is my desk neighbor, we can collaborate on how to make it integrated with Pynapple. So what we teach usually in a workshop is that you start from the NWE file, the data, you do some data wrangling with Pynapple, and then you do the spike counts. Basically, you bin the spike train, you get your feature and the right time axis, and then you usually use NEMOs to do some neural modeling. So right now, what it implements is mostly the GLM, basically a linear, nonlinear Poisson process model, where you basically have a feature matrix here, where this is the time axis. Those are your features. You're going to learn a set of weights for which you're going to basically predict firing rate, okay, that's going to be best describe your spiking activity. That's how you can describe what NEMOs does right now, all right? And this is done in a few number of lines. Of course, this is reminiscent of scikit-learn. If you use scikit-learn, this is the idea. You instantiate the class, you fit your data, and you predict, and you can get a score of your data, okay? So we have a lot of examples online of how to fit neural data, starting from like NWB to Pynapple to NEMOs, all right? And more than just having fitting like a GLM, there is also a whole module for basis in NEMOs. Basis are a good way of, how to say, lowering the dimensionality of your problems. So instead of basically trying to fit your model from the raw feature, you can represent your feature as a set, a limited number of bases. The example here, for example, would be a 2D position of a mouse in an arena that could be represented as a set of bumps here, from which you can weight them, and from the weights, you can learn the firing rate of your neurons, okay? So you can use this as predicting the activity of your neurons and so on. There is even more than that in the tutorials. We also show how to basically predict the activity of your neurons based on the activity of other neurons to derive some coupling filters. Okay, so you go online, go check those tutorials. If you have some data sets with and you want to see how, for example, some people mentioned yesterday actually cross-region talking or coupling, and we have some tutorials about that where we show how some neurons can have some coupling by basically predicting an activity of a neuron based on the other neurons. Okay, so go check that online. And what do I want to say here? This is Eduardo's slide. So, okay. So this is NEMOS so far from Pynapple in to learning a GLM model all the way to Pynapple out. NEMOS is also smart. It can spit out if you pass Pynapple TSD frame as your feature, it will spit out a Pynapple TSD frame as your predicted firing rate. Okay, so you can align in time. And this is how it's done here with the call of a few functions. So I will not describe that too much. You can go online for the example, from streaming from Dandy to doing some pre-processing with Pynapple where you bin the spike train, you interpolate to create the features, all the way to creating the basis functions, to computing the feature, to fitting the GLM, population GLM, okay? So all of this is online. So right now it contains a Poisson model for the observation model, gamma, negative binomial, Bernoulli. There is regularization model, lasso, group lasso. There is a bunch of solvers. This list is always changing because I forgot to say that this depends on JAX, and JAX is very, very evolving like a package where nothing is really stable, but at least the API is stable for the user, but under the hood, the backend is kind of changing rapidly when it comes to fitting the model. But you have all those possibilities right now. There is a basis module. We implemented a bunch of different bases, raised cosine linear basis, if you are familiar with the work of Eros Simoncelli, M-spline, B-spline, and so on. And it's compatible with scikit-learn, okay? So that's NEMOS, kind of the companion package of Pynapple when you want to do data analysis, all right? I would like to mention that we are hosting workshops. So the next workshop will be at SFN in San Diego. The idea is that you come for 2 days and we teach you Pynapple and NEMOS. So we provide the data and everything. And we provide the environment, you install the environment. And for 2 days, there are talks on like practice, hands-on data where you basically learn how to analyze data with Pynapple and fit a model with NEMOS, basically. 2 days, next one is San Diego. SFN, okay, so you can now register. Or you can, maybe if you have some member of your lab who wants to learn about Pynapple, you can indicate them about this workshop. A bunch of links, and I will answer Ben's question now. So there is a package that is not done, and I will do like what you guys did, like you can be the beta tester of this, like your notebooks yesterday. So since you have, if you want to try something funny, if you want to try something new, you can try Pynavis. Because Pynapple is basically a bunch of time series that all share time axis. Of course, the natural things to come in mind is aligning, visualizing data aligned in time, basically, okay? So of course, like you can have like your spikes on your LFP, and if you have tried to do this in Matplotlib, it can be very painful to browse through data that have like the different sampling rate or different axes and so on. So the idea would be to provide some sort of tools for people to do this automatically. So I'll show you a demo now so that you can see it. Do you have time or not? Yes. Yeah. Okay. So the idea, and you can install Pynavis yourself and try it. Actually, I will try with the IBL data here. I was testing that. So the idea would be that you can basically synchronize streams of data. And here in this example, I'm going to make an example where I have 3 videos, basically. I don't know if it's going to still work. Let's see if it still works. Oh yeah, still. So here, in the IBL data, they have this kind of recording of videos where they have like they have a mouse doing a task, and those are 3 different movies saved on disk, basically, with different sampling rate. And the idea is that now, if I scroll through one, it's synchronized automatically in time, basically. So as you can see here, the mouse is moving. And it's moving in time for all 3 movies, basically. So that's one possibility you can do with this kind of package, basically, where you define this kind of object, and you can scroll through time, okay? That's one example, all right? I'm going to show you another example. I'm going to show you this one. A bit more complicated. So here, now I'm gonna put together different type of data, some multimodal data sets, which is something can be very annoying to do. Here I have some spikes. Here I have basically, I did a manifold projection of my spikes, so I bin my spike train. I do the isomap projection in 2D, okay? So I have a 2-dimension here. And here is a video of the mouse doing a task. So basically you can see the mouse is just running around. And the idea is that basically as you scroll through time in one axis, you scroll through time in all of the other subplot, basically, all right? So then you can see, this is about data exploration. This is not about creating final figure visualization. This is about, okay, I got my session, I got a good session. I want to see if my data are correct. I want to see if what I'm gonna, like I want to do some data visualization where I want to see if all my time are properly aligned, basically. Am I detecting some event crazy here? And I want to see if this match with something, another particular feature, basically. And so here, as you can see, but here, of course, this is a very, I will describe this data set. These are head direction neurons. These are neurons that fire for a particular direction of the head of the animal in the horizontal plane. So whenever the animal is facing this particular direction, those neurons are firing. And now we're going to see, as you move around, the animal is going to move his head, and you're going to see that the firing changes. Let's find an example. Yeah, you see here, okay, now it's going to move, and you see those neurons now fire. Those are head direction neurons, okay? So they change their firing based on the direction of the head of the animal. This is a 2D representation, like a manifold representation of this firing. As you can see that this represents basically the head of the animal in the horizontal plane. So this is a type of visualization you can create. And the idea is more like, okay, I want to do some data verification, data quality, by plotting multimodal data sets, basically. That makes sense? Is it what you wanted to see? Okay. I will show a bit more. Do you still have time? Sure. So one annoying things to do is like I don't want to script that. I spend enough time in my life scripting my Plotlib stuff that I want to do it ever again. So the idea here is that actually you need to do it. We're never going to beat Matplotlib. Matplotlib is too good, or any other packages. I don't want to beat them. What I want to do is that I want to load the session and quickly check that my time is properly aligned. So here, for example, I want a GUI that gives me this type of visuals automatically, and that's the idea behind Pynaviz. And for example, I have so these are just random data. I have some time series. I have a TS group, and automatically the time is aligned by this, basically. So then you create this kind of visuals where you create those called widgets, and those widgets basically automatically synchronize in time, and you can check your data like this, basically, so that you can see. It's basically a data quality. All right, makes sense. So this is kind of this will be the companion package of Pynapple when it comes to data visualization. All right, and then of course I will just show you one last example just to explain the whole capabilities. Because I talk about metadata, and the cool thing about this is that you can also leverage the use of metadata. So I'm going to show one more example. Here I have some LFP. So this is some LFP data from the disk. Okay, so here is reading from disk, and I plot the first second. And I have associated with the TSD frame object the channel and the group order, basically. So what I can do is that I can say group by, and I can say group by by group, actually. And it's going to split by groups. I can also say sort by, and I want to sort by channels, basically. And I sort by channel. And I can say also color by, and I can say, for example, color by channel, actually, or by group. Yeah, okay. I can show something better. Yeah. So here, basically, this is possible because I have metadata associated with each object. So it depends on you of putting the right metadata. But once you have this, it's easy to visualize any time series synchronously on your laptop, basically. And this, of course, will only be on the laptop, and it's not for like offline or online use. It's only for local use. Okay, so that's the idea behind Pynaviz. Unfortunately, it's not finished, and hopefully it will be done soon. But if you start using Pynapple, stay tuned for the release of this package because it can really help you basically visualize your data. Okay. Any question about this? Make sense? Is it no? Okay. So coming soon, hopefully. All right. And I think I'm done with this. Yeah, I'll just close this. Okay, any question? Yes?
When you showed the
Nemos, yeah.
There was some like streaming of data from Dandy.
Yes.
Is there any other way to stream directly into Pynapple, or is it? Yeah, how does the streaming work?
Yeah, so here, how to say? So Nemos has this kind of little module called fetch, which under the hood is Pooch. Do you know Pooch? No, it's something that basically can stream the data. So I think this is and Nemos can do it. Yeah. But you can do also use the Dandy application, like the Dandy app also can do it, right?
Okay, so
This is very, very little part of Nemos. So this is a very little part, yeah. It's not, I don't know if that makes sense. Any question? No. All right. Good. Thank you very much.
Okay.